So our first presenter that I'm going to introduce is Dr. Carl Bradley. He wants everybody to know that he used to work at NDSU as an extension plant pathology specialist. He says he was the guy that followed Dr. Art Lamy and preceded Dr. Sam Markell. And Sam will be the other presenter that I'm going to introduce in a second. But Dr. Bradley's current job is as a professor and extension plant pathologist at the University of Kentucky. He's based out of a research and education center in Princeton, Kentucky, which is in the western part of the state, which is close to southern Illinois and southwestern Indiana. He's been there for about 6 years. He's originally from southeastern Illinois, a town called Ridgeway, Illinois, and actually got his higher degree, PhD, at the University of Illinois at Urbana, Champaign. He has a statewide extension and research responsibility for disease management of soybean, small grain crops, and winter canola in Kentucky. His main focus in Kentucky has been on fungicide resistance and pathogens of soybean and wheat, and management of important diseases and pathogens that occur there, such as frog eye leaf spot of soybean, SCN, and Fusarium head blight of small grain crops. Our other presenter is Dr. Sam Markell. He is the North Dakota State University's professor and extension field crops plant pathologist. He's worked at NDSU for 13 years, and he puts in parentheses, ever since my predecessor and co-speaker Carl Bradley found greener pastures elsewhere. He says his native state is Minnesota and has a PhD degree from University of Arkansas in plant pathology. And he has an MS and a BS from NDSU in plant pathology and biology, respectively. His responsibilities are to develop and deliver management information for diseases of broadleaf crops grown in North Dakota. So these include sunflower, soybean, dry edible bean, canola, flax, pulse crops. And his focus has been on applied disease management and monitoring pathogen population changes. Recently, he has been co-leading the SCN Coalition, which is a public-private partnership whose objective is to increase awareness and mitigate yield losses caused by SCN. And take it away, gentlemen.
Thanks, Angie. And yeah, I was the blip on the radar between Dr. Art Lamy and Sam Markell, but it's good to be back and be able to present to everybody. So Sam and I are going to try to co-present this, and so hopefully we have it figured out. But Sam, I'm going to pass it over to you, and you should be able to advance slides, I think.
All right, good, Carl. Thanks. Yeah, welcome everyone. Thanks for joining us. I'll try to control your screen, okay, Carl. And thanks for vacating that job. I really appreciate it. So today we're going to cover SCN, sudden death, and brown stem rot. The reality, I think, of what we're going to discuss is the sudden death syndrome is probably going to be the main course. Soybean cyst, we need to talk about a few things, and we'll show you a few updates. Probably not talk terribly much about brown stem rot, but throughout, you're welcome to ask questions about any of this. So I'm going to start with SCN, and then Carl will take over from there, talk about SDS. So just to put everyone on the same page here with SCN, and many of you are aware of this, it's a parasitic worm, it's invasive, continues to expand, it's soil-borne, and it moves with any way that moves soil. It can cause problems in all soil types, tends to be a little more severe in lighter soil, and high pH really increases reproduction, so that's been a problem for us. Probably goes without saying, it's the top yield limiting pest in soybeans, and that is becoming that way in North Dakota, Minnesota as well, and it's overcoming genetic resistance. So This is just a visual. You can see a nodule on the lower left corner of your screen, and you can see SCN white females. So these are essentially cysts full of eggs everywhere else on that root. And I took this picture this summer. Pretty severe infections are not uncommon, especially in the southeast part of the state. So ironically, Carl Bradley took this picture in 2003 in the first report of SCN in North Dakota. And that's Dr. Berlin Nelson there almost 20 years ago, digging up some roots. So it was the first introduction. It's been a while. And you can see that when you get pretty severe damage, you get some general yellowing, but it's not very diagnostic. And many of you know that. Just to reiterate that this thing really does spread effectively. It was first found in the 1950s, initially North Carolina, but it made it to the Boot Heel of Missouri by 57. And it is spread consistently throughout all the soybean producing area in the U.S. There was a press release just last week showing the updated map of distribution. And so the blue counties are all new finds since 2017. The red counties were positive prior to that, and then the counties with no color and no SCN has been reported or detected. You can see this thing is still continuing to expand, notably in North Dakota, northern Minnesota, and New York. And if you have good eyes, here I'll help you, you can see that there's 4 areas in Manitoba now that have some SCN. So it's going to continue to expand. Now, just because it shows up as being here doesn't necessarily mean that it's everywhere in the county. Many of you are aware of the North Dakota grower-based sampling program that we've been doing since 2013. And this program is set up so that the North Dakota Soybean Council covers the cost of lab fees for growers that want to sample. And they just pick up these pre-marked bags at the county office or through me or Soybean Council, send them in, they get their data back in the mail. I think what's important here is 2 things. The first is that, and I should define this, the black circles are negatives and the gray boxes are kind of inconclusive, really low levels, but anything that's got colors in it is going to be real, right? So the first thing that's important that you'll see is that you've got a bunch of blue circles and green triangles starting to show up outside the southeast and east central part of the state. The other thing that's important is that the southeast part of the state has some incredibly high levels, and we've got a lot of data points there. And so we put this together in a heat map just recently, and you can see that yellow is 200 to 2000, but these color shades of orange and even into the red, so they're getting pretty high. At these levels, you can take yield loss. If the environment's right, you can take it pretty bad. So you have to manage this pretty much everywhere in this part of the state. And I would suggest that growers would benefit from sampling these areas of known infestations to see if their management tools are actually working. So we've got a little data to share with you on that. So the last time I was here, I shared this slide with you, trying to emphasize that genetic resistance is really good when it works. So how you read this is you've got yield on the left, you've got 4 locations, you have a susceptible check, so soybean with no resistance, and then you've got 30 to 50 different resistant varieties. Commercial, this is Roundup Ready, Liberty Length, there's in separate trials, but it's all the data compiled here, right? And so you can see the value of resistance over a susceptible, it's about a 40% yield increase when SCN heats up on you. But that only tells part of the story. And I'll show you another slide, but just keep in mind here that Galesburg SCN did not show up here in Galesburg's, right? So there's 3 of the 4 sites. Here are individual soybean varieties. So you can see here, you've got this yellow bar as the one without the genetic resistance, and you've got a bunch of soybean varieties that are no better than that susceptible. And then you have kind of this steady grade of better efficacy. And a lot of that's due to the genetic resistance. So we've known for a while that not all resistant varieties are created equal. And that's because in the breeding process, this 88788 that's in most of our varieties, there's lots of little genes in there. And some of those get sloughed off in that process. So you might have a variety with 88788 that's really effective and one that's not so effective. Well, recently, Dr. Guiping Yan started to look at some of this in the greenhouse, and these are pretty laborious tests, and she's done this over the course of a year. And what she's done is taken 45 to 50 varieties, and she's looked at the SCN reproduction on all these varieties. And these are all varieties that are sold that have a source of resistance. They're almost all PI 88788, but there was a couple of Pekings in there. And based on her reproductive work in the greenhouse, which is the standard way we would say it's resistant or moderately resistant or susceptible, she's found that the majority of varieties sold with genetic resistance 88788 or Peking are actually susceptible or moderately susceptible. So the message here is that when you get a variety with 88788, it may in fact be resistant or moderately resistant, but it also may not be all that much better than the susceptible. And this is all based on greenhouse reproduction. I would emphasize that any of these, well, any of these, but a susceptible, are still offering some control of the nematode, but they're not what we would have hoped they were. And so this is a problem in our area. It's also notable that the pathogen, the nematode itself, is starting to change, and we're starting to see it adapt to 88788, which is unfortunate. So it means it's harder to manage, essentially, with the tools we've got. I want to just introduce you quick to the SCN Coalition for a little more information if you haven't heard of this. It's a public-private partnership, and it's really well funded by the companies and the national, regional, and the local checkoffs. There's a lot of universities involved. And we've gotten together to try to unify the message around SCN, to really try to help growers and everyone else that works with soybeans understand that the nematode is changing, and managing it takes a real active strategy. It's just more complex. One of the newest tools that we have made available is this Let's Talk Toads program. And I would encourage you to visit with your growers about that. There's 23 different videos online right now. They're all 2 to 3 minutes in segment, and they're all organized in a way that it answers a certain question. So like on this page here, you can see how does weather impact SCN? And then there's a 2.5 minute video clip, and it's talks specifically about weather. It's notable that we filmed in 4 different states, so there's information that's different depending where you are. In North Dakota, we were at Mike and Chandra Langsa's farm in Richland County talking about SCN. In Iowa, there's more videos, and they're looking at different seed treatment trials and genetic trials, and then visiting with a grower down there. So I would encourage you to take advantage of that. So I think I'm going to pass the ball to Carl here.
Sure. Yeah. Thanks, Sam. Interesting stuff. It's interesting to see how SCN has really spread across North Dakota. When I was there, we found it just in Richland County back in 2003. And gosh, things almost 20 years later, things look a lot different. And so Sam had asked me to come on and speak mostly about sudden death syndrome, which I understand is now a problem in North Dakota as well. And this is a picture I actually took from Southern Illinois. I used to be at the University of Illinois prior to coming to UK about 6 years ago. And you can see the patchy distribution there. It's a soil-borne organism. So oftentimes when we're dealing with things below the soil, they're going to be just sort of patchy in the field. They're never really going to be uniform. Sam, you might want to talk about this one.
Yeah, I think Carl will get into symptoms and movement and stuff as well, but I think it's good for everyone to put their head around where this thing has been found, where it is. So this is a slide that Dean Malvik at the University of Minnesota sent me yesterday, and it's basically showing a progression of the pathogen and then the disease north, and it's not a surprise, right? But in our area, it's in Clay County, it's in Hubbard County, and it probably is also in additional places. So this is not something that's just a Southern Min problem anymore. And it was for a while, but it is not. In North Dakota, it's a similar scenario. We first confirmed this in 2018 in Richland County. That was really no surprise. 2019, we didn't see much of it. In 2020, it really blew up in Richland. Unfortunately, we had a second confirmation in the state, and that happened to be in Cavalier County. And that was a surprise. It was an unwelcome surprise. It was definitely SDS. There's no doubt about it. We confirmed it multiple ways. Ironically, we couldn't find SCN in that field, but those are intimately linked. And Carl will discuss this. And I'm just going to overlay the map here where we know SCN occurs. So I'm going to suggest to everyone here that just because you haven't seen SDS in your field doesn't mean you should look for it. And I think the Cavalier find kind of proves that. So I think everyone should keep in mind that they are at risk, at some level of risk for this. So keep your eyes peeled. And this, I would say you can take the Red River and you can flip this map over, and the same issue, same risk would be on the Minnesota side. Go ahead, Carl.
Yeah, so we kind of mentioned the distribution that you might see in the field. I think this is one of the pictures that Sam had taken. I'm not sure where that's at. Richland?
That's this summer in Richland County.
Okay. So one of the first things that you might notice with SDS is the what we call this initial chlorotic flecking. And so you can just sort of see the little yellow flecks that are on the leaves. So that's going to be one of the first things that you notice. We don't normally, at least in Illinois and Kentucky, we don't normally see these symptoms until the plants start to switch over to reproductive phase, usually around flowering, or maybe a little later, even around pod development, maybe when we see these symptoms start to occur. But it really probably depends on susceptibility of the variety and conditions and lots of other things. So those little flecks are going to spread, and they're going to form this interveinal chlorosis, or the yellowing between the veins. That chlorosis is going to turn into necrosis, or dead tissue, again, interveinal. So it'll be in between the main veins that you can see here. Okay, so we see a lot of these leaves are really just about fried, but those veins remain green. So that's one of the symptoms that you're going to notice with SDS. The other thing that we notice is those leaves are going to fall off prematurely, but those petioles usually stick around a little bit longer. So you'll see a lot of plants that are affected that have lost their leaves, but those petioles remain attached. And then this is one of Sam's pictures. Sam, you want to talk about this one?
Yeah, so one of the things that we do when we're in a field, and Carl admittedly has quite a lot more experience than I do, but I always bring a pocket knife, I bring a hand lens, and I bring usually a diagnostic hard set or something. And if we see foliar symptoms like Carl described, it could be a couple things. It could be brown stem rot. I think, Carl, you've got a couple photos of that. I always take and I always cut the stem open. And if I see in the center of the stem, the pith all brown, where it kind of looks like lead in a pencil, it's brown stem rot. But sometimes with sudden death, you can see some browning if you kind of shave off the outer tissue and kind of slowly peel it away. You'll see browning and not in the pith tissue, but the rest of the cortical tissue. And that is something that you'd sometimes see with sudden death.
Yeah, and we noticed that usually when you see that, it's usually towards the lower part, usually in the root, but it might be just like above the soil line, but usually not much further past that point. The other thing you can find if you carefully dig, and I know some of those heavy Fargo clays is kind of hard to be careful when you're digging and gently taking off the soil. But if you can do that, you may notice that in infected plants that you may have this sort of bluish, whitish colored group of spores that are going to appear there on the root. So you can see that I kind of have the close up there. One thing to note is, at least from my experience, you don't always see this. And in fact, it's a little bit more rare to see this. I would say I see this less than 50% of the time that we have SDS out in the field. However, if you do see this and you also see those symptoms that we just described, very likely it's SDS. But if you see those symptoms and you cut the stem open like Sam talked about, and you don't see that browning of the pith, but you don't see these spores on the roots, it could still be SDS. I just wanted to talk about the disease cycle here. This is a soil-borne pathogen. That's a Fusarium. It lives in the soil. It can overwinter on soybean tissue. The interesting thing about this is that it also can live off of corn residue. Dr. Dean Malvik at the University of Minnesota has published some work looking at a lot of potential hosts and found that there are a lot of asymptomatic hosts of this pathogen. So when we talk about crop rotation, that's kind of a big question mark because there are so many hosts that this fungus can potentially survive on. So as early as those plants start to germinate, so the radicle emerges from that seed, that you have the young seedling root, that's about as quickly as the SDS pathogen can infect. So as soon as those roots start to emerge, you can have infection. Now the fungus will stay down in the root zone, and it will grow around the root, and eventually it'll make its way into the cortex, and then into the tissue that moves things around, the xylem and the phloem, usually the xylem, and it'll produce a toxin. And that toxin then is moved up through the xylem, up into the upper part of the plant. So you can kind of see the diagram here. That toxin will accumulate in the leaves, and that's why you see those leaf symptoms. Now, even though you're seeing those leaf symptoms, the whole time, the fungus is actually down in the root zone. So the fungus doesn't travel up with the toxin. So if you bring in samples to the diagnostic lab at NDSU or University of Minnesota, be sure you bring in entire plants, and especially that you have the roots involved. Because if you're just bringing in leaves or just the top part of the plant, you won't be able to. The diagnosticians won't be able to find the fungus.
I would add that's a really important thing for our region. Not a lot of folks know that. I think we had a lot of calls this summer, and we saw multiple samples, and most of the time it was stem and leaf tissue. So the roots are absolutely critical for us to be able to identify it.
Yeah, actually, this disease first started occurring, back in the 1970s, and it took a while before pathologists were actually able to document what pathogen was causing this. And that was part of the problem, as they started looking in the leaf area, and so it took a while to sort of figure out what was happening there. But yeah, the other thing to mention here is the type of conditions in which this is more favorable. We find that if you're planting in the cool, wet weather, and if you live in North Dakota, that's pretty much probably when you're planting, based on my experience living there at least. So cool, wet weather, especially after planting, is very important for pathogen infection. At that time, that radical's growing very slowly. It takes a while for the seed to germinate, and that gives the fungus a lot more time to penetrate and get into that root. And then if we have adequate moisture throughout the growing season, especially June through August, that really makes the toxin move up, and we see a lot more severity. So cool, wet weather at planting, and then adequate rainfall through the rest of the season. Usually we talk about high yield environments. So here where I live now, if we have a year where we are really just pushing yields, really having a good year, we're likely to see more SDS. So high yield environments usually go along with more SDS. Sam mentioned the interaction between SDS and soybean cyst nematode. There's been a lot of research in this area. It's actually a pretty complex relationship, but the bottom line is you don't have to have soybean cyst nematode to get SDS. However, if both of those pathogens occur in the same field, you have a higher risk of SDS being even more severe when SCN is also present. And it sounds like now that you have both pathogens, then, and especially in the Minnesota side as well, you really need to think about managing both diseases since they're both present. There's a lot of different research that's been looked at as far as managing this pathogen. Some of the cultural practices that have been looked at since the 1980s have included crop rotation, planting dates, lots of different things. I mentioned crop rotation may not be as effective because of the ability of this pathogen to grow on a lot of different potential hosts. Planting date is one thing that can make a difference because of the soil temperature. So we know if we plant in that cool soils, that the risk of SDS goes up. But that's usually one I don't talk a lot about to farmers, because I understand when you have the window to plant, you really need to go plant. And so that's one we know that planting date can make a difference, but usually if you have the window, you really need to go plant. In our area, we do have genetic resistance. It's been around for quite a while. Probably when you're getting into some of those group zeros, double zeros, most likely the seed companies have probably not intentionally bred for it, but maybe there's some varieties that could have some level of resistance. And then we've got some seed treatments that we've been able to, that have been registered over the last 5 years or so. I'm going to talk about some results from some research trials, from university research trials. Just the things to note, this is a hard disease to work with if you're just going into like a farmer's field, because it's very erratic on how it is in the field. It's very patchy. So we do some things in our research to be sure that we get some uniform SDS. So one of the things we do is we plant early. Here in Kentucky, that would be in early April. We inoculate with the SDS fungus during planting, and then we use drip tape, and we apply 1 inch of water per week, just about as soon as those plants emerge. And you can see we have 4-row plots, and you can see them, and we just put the drip tape in the middle 2 rows. So that's what we evaluate and we harvest. But you can really see how the irrigation makes a difference. So here's row 1, and then rows 2 and 3 here are affected. And you can see the same thing over here as well. So I'm going to show you some results from some this one's from a multi-state trial that I took part in when I was at the University of Illinois. This was conducted in 2015 and 2016 across 6 states, and then Ontario, Canada was also included there. What you're looking at, so this is over 2 years, so 2015 is on the left and 2016 is on the right. So what you're looking at is the SDS severity index. So the higher the bar, the more disease there was. We had some different seed treatments and then foliar products. So what we're calling the base here, that's just your base fungicide, insecticide, and then all of the other treatments included that same base, and they just had that next treatment on top of it. Then we had Olevo, which is a seed treatment. We had Heads Up, which is a seed treatment, and then Cobra, Fortix, and Procytic are all foliar products that we evaluated. And you can see in both 2015 and 2016, the only place that you see that asterisk is over that Olevo bar. So out of these products that we evaluated, that was the only product that statistically reduced SDS severity. We also, when I was at the University of Illinois, we also had a planting date by seed treatment by variety trial. And so you can see that we had a susceptible variety over here on the left, and then a moderately resistant variety over on the right. You can see our different planting dates that range from April 15th to June 15th. And then looking at the SDS index, you can see we had a little bit more, well, a lot more disease in our susceptible variety overall when you just look at the left graph versus the right graph. But anywhere we had included Olevo, we were able to significantly reduce the level of disease in our susceptible variety. As well as the moderately resistant variety, even though we didn't have a whole lot of disease to start with. And the other thing to note is, you know, the earlier the planting dates, we tended to see the trend of having more disease the earlier we planted it. With some exception, there was a little bit of a jump here in our June planting. And so when we look at this is yield, we can see that overall our moderately resistant variety had a much greater yield than our susceptible variety. And especially in our susceptible variety, because of the level of SDS, we were able to see a yield response in most of those planting dates. And if you look at soil temperature, this are different, sort of the calendar down here, and these arrows represent the dates in which the different planting dates were. And you can see how our first planting date was under 60 degrees. It was right around 55 degrees or so. So pretty cool soils that the seeds would start to germinate at that temperature, but it would be really slow. And temperatures climb back up, but it was interesting to see that June planting date where we had a little more SDS, and you can see for whatever reason we had a little bit of a cold spell during that period, and it did drop the soil temperature a little bit. And so I think that was part of the reason why we did see that spike even in that June planting. A little more recent results. This is from some trials here at Princeton, Kentucky, where I am now, from 2018 and 2019. This is 2 years combined at one location, 2 years combined. This is SDS severity again. So the higher the bar, the more disease. Here's our base. Here's a new product from Syngenta called Saltro, which has efficacy against SDS. And you can see that we reduced the level of disease with Saltro. And then Chloriva, which is really a product just for soybean cyst nematode, the combination of Saltro and Chloriva, and then Olivo at the 0.15 milligram rate. And you can see that both Saltro and Olivo were pretty similar here in regards to reducing disease. As far as yields, I'm proud of this slide because we got close to 100 bushel soybeans here. Keep in mind, though, that we do irrigate this. And so this is not typical for Kentucky soybean yields. But if you irrigate 1 inch of water per week, you might get yields like this. But the thing to point out is that we did have a significant response to some of the seed treatments relative to our base treatment because of the level of disease and the level of protection that we were able to get with the seed treatments. So overall, what I tell our growers, at least where I live, is we start with a variety with a high rating for SDS resistance. And I understand that's something that you probably are not going to be able to do right away until you know more about the level of susceptibility in a lot of the varieties that you grow. The other thing I tell our growers here in Kentucky is if you're going to plant early, if you're a farmer that likes to go out and plant soybeans about as soon as you can get out there, as soon as it dries up, early April for here, and you know that there's a risk of the temperatures really dropping, that you should really consider using a seed treatment like Olivo or Saltro because of the risk of SDS in our area. And I also tell them that these seed treatments are not bulletproof. You saw that even with the moderately resistant variety in the one trial, with the susceptible variety with the one trial, even though we had the Olivo added, we still did develop disease. So these seed treatments are not going to prevent you from getting SDS, especially if it's on a susceptible variety and the conditions are really favorable for SDS. And what I tell our growers here, the best scenario, use a most resistant variety. And again, if you know you're going to be planting early, pushing those planting dates, consider one of these effective seed treatments on top of that. Sam mentioned brown stem rot. I just wanted to mention that it has the exact same symptoms as far as the foliar symptoms for SDS, but Sam talked about splitting those stems, and this is what you're more likely to see with brown stem rot versus with SDS. If you're looking at that pith, you're going to have a healthy pith there. I just wanted to mention funding. Some of the work that I did when I was at Illinois was sponsored by the North Central Soybean Research Program. We have a lot of industry partners that help fund our research as well. And Sam mentioned that the SCN Coalition, that's a really good place to go and get information. Andrew mentioned in his presentation the Crop Protection Network, and I'll mention that here as well. There's some good information on seed treatment efficacy. There's a seed treatment efficacy guide there for soybean. And then there's also a nice multi-state publication on sudden death syndrome. And finally, that's it. I also, just to show that I actually was at North Dakota, this is me, gosh, almost 20 years ago. Marcia McNamolin took this picture, and that's me standing in a flax field wearing an NDSU cap. That's the only picture I could find with me wearing any NDSU garb. But anyway,
look like you're about 12 years old, Carl.
I know, man. I don't look quite like that anymore, but sometimes I feel that young, but then sometimes I don't.